This is a A-level presentation looking at the structure of RNA and the process of transcription as part of protein synthesis. At this point, you should pause the video, take time to read through what's on the screen in front of you, come up with your answers, and then when you're ready to check your work, you can restart the video. So pause the video now. The genome refers to the complete set of genes in a cell. The proteome is the full range of proteins that a cell is able to produce. A codon refers to a triplet of cells or three bases that occur on a messenger RNA, and an allele is the different versions of a gene. You're also asked to draw and label a nucleotide. When you're drawing and labeling a nucleotide, there are three main parts. You have the phosphate group, the pentose sugar, which depending if it's DNA or RNA will be ribose or deoxyribose, and then you'll have a base. And again, depending on if it's DNA or RNA, you have slightly different bases with thymine and uracil. In terms of DNA and RNA, DNA is double-stranded. It has a pentose sugar of deoxyribose and the bases are A, G, C, and T. You don't have to use the full names, the letters are absolutely fine. And with RNA, it is single-stranded. We have a ribose sugar and we have the same as in DNA. We have A, G, and C, but we have uracil instead of thymine, so U instead of T. And the last extension question was, can you name three types of RNA? And the three main types you need to know are messenger RNA, transfer RNA, and ribosomal RNA. So what you have in front of you, at first glance, you may think is a double-stranded DNA molecule, but in closer examination, you'll actually see that the left-hand side, you have a repeating pattern in the chemicals. And the repeating pattern is the sugar phosphate backbone of a nucleotide strand. On the right hand side in the orange colored box, you have each of the bases that is attached to each of the nucleotides. And as a consequence, this is actually single stranded. The other thing that's there is the fact that there's actually no thymine there, there's actually uracil. And what we have there is a single stranded messenger RNA structure. Again, as previously said, we've got ribose sugar, whereas in DNA you've got deoxyribose sugar, and we talk about triplets, and triplets are any set of three bases. But when we talk about a codon, we're specifically referring to a set of three bases that is found on a messenger RNA strand. The second type of RNA you need to be aware of is transfer RNA. Transfer RNA, as you can see, is single-stranded, just like messenger RNA, but it forms a more complex structure. The main parts of that structure that you need to be aware of for A-level biology are at the very bottom and at the very top. At the very bottom, we have what's called an anticodon loop and an anticodon. The anticodon is three specific bases that are complementary to the three bases that can be found on a messenger RNA codon. Each transfer RNA has its own specific anticodon, and each transfer RNA is complementary to a particular codon on a messenger RNA. At the other end of the transfer RNA, we have what is called an amino acid attachment site. At this end of the transfer RNA, we find amino acids. And again, each amino acid is specific to a particular transfer RNA, and hence each amino acid is specific to each anticodon. So each anticodon will always give you the same amino acid. Before going on to the process of transcription, you need to make sure you've memorized and you are secure in your knowledge of those two types of RNA, which would be the messenger and the transfer, because the next sets of processes which will lead into transcription, and then eventually that will lead into translation, and you're going to find it difficult to follow those concepts if you don't know what we're talking about when we mention messenger RNA and transfer RNA. So make sure before you go any further, you have taken the time to memorize the first section of the presentation. In genetics, basic premise is that DNA can be used to produce RNA, which can be used to produce proteins. The process of producing RNA from DNA is known as transcription, and the process of producing protein from RNA is known as translation. Remembering that proteins are made up of polypeptides, and polypeptides are made from amino acids, and proteins can be from one polypeptide or several polypeptides. There is a question they're asking about 4 levels of protein structure. The 4 levels of protein structure are primary, secondary, tertiary, and quaternary. And you do need to know the definition of each of those, and you do need to know the bonds that are associated with each of those different terms. In primary structure, we talk about the sequence of amino acids. In secondary, we talk about the folding in terms of alpha helix or beta pleated sheet. In tertiary, we talk of how the polypeptide folds into a 3D structure. And in quaternary, we're talking about several polypeptides joined together. Often hemoglobin is the first example of this that you'll cover in the course. So to the process of transcription. What we have in the image is a DNA double-stranded molecule, and we have to use an enzyme to unzip that double-stranded molecule of DNA. The enzyme we use is an enzyme called helicase. And what the helicase does is it breaks the bonds, the hydrogen bonds between each of the base pairs, and as a result it exposes a particular sequence of DNA, often a gene. Now, in the A-level biology course, you should refer to the particular section of DNA that is being transcribed as the template strand. But you may find in some older questions and some older textbooks that they refer to what's called an antisense strand, an anticodon strand, or a transcribed strand. Now, these terms are perfectly acceptable in terms of they are accurate, but the most recent and the term you should memorize and use, and the term that will appear in your exam, is template strand. And the template strand simply refers to the section of DNA that we are going to use to produce our messenger RNA. So what we've got on the screen now is the DNA strand, that is the template strand from the previous picture, and we've just put it into a table to make it easier to follow. So at this point, I suggest pause the video, work out the complementary DNA strand, then look at the messenger RNA strand and work out what that sequence would be, and then you can go back and look at the original complementary DNA strand, and if it had been the template, what would the messenger RNA strand look like? So pause the video now, have a go at that task, and then when you're ready, you can restart the video. So the complementary DNA strand, we simply work through each of the letters on the template and put in the complementary version. So A, we go T, T is A, GC, CG, A, T, C, and G. We then take the same template and we're producing a messenger RNA strand, and what we do is we have exactly the same as we've done in the first one for the complementary, except that all the T's that we put into the complementary strand we would write U's in the messenger RNA strand. So a good way to avoid confusion is when you're translating or when you're doing DNA replication or any of those processes where you have to work out complementary DNA strands or RNA strands, I would always suggest make the DNA strand first, no matter what the question says, and then read the question and check. And if you need to produce an RNA strand, you can simply cross out all the T's and replace them with U's. So as a consequence, you would end up with U, A, C, G, A, U, C, G. And then what you've got at the end is the extension question, and if you were to take the complementary DNA strand to make that into a messenger RNA, you would end up with A, U, G, C, U, A, G, and C. And again, all we would do is first write out the complementary DNA and then cross out all of the T's and replace them with U's. Now, when we're producing messenger RNA strands, there is one other process that applies to eukaryotic cells. This does not apply to prokaryotic cells. Prokaryotic cells will just do the previous process. However, in eukaryotic cells, we have these things called exons and introns. Now, I don't know why, but for some reason, everybody thinks introns are the ones that code for polypeptides. But they're not. It's the exons that code for the polypeptides. So introns, the letter I for introns is important, but the E for exon is because they are extremely important, because those are the ones that are coding. As a consequence, when eukaryotic messenger RNA is produced, we start with a DNA strand, and we have the RNA polymerase, and the RNA polymerase sticks all the RNA nucleotides together, but it copies the introns and the exons, and as a consequence, we end up with what is called pre-RNA. When we have pre-RNA, we then have a process known as splicing, and in splicing, we remove the introns so that we are only left with the exons in the messenger RNA. I have seen some exam questions where they literally give you, like the previous slide, where they give you a lot of letters with the nucleotides, so for example A, T, G, C, etc., etc., and they ask you to write out what the messenger RNA would be, but they also identify some of the bases, some of the triplets as being introns, and some of the triplets being exons. And when you're producing messenger RNA, it's only the exons that would be copied across into the messenger RNA. So just look out for that question. The overall process is the DNA is transcribed as known as the template. The exons are the ones that are coding because they're extremely important. The introns are known as non-coding regions. We have the pre-messenger RNA is copied from a DNA strand by RNA polymerase. Remember, polymerase will stick nucleotides together. If you want to make another DNA strand, you'd use DNA polymerase. In this case, we want to make RNA, so we use RNA polymerase. Again, we've got the complementary pairing of our bases. Remember, in eukaryotes and prokaryotes do not have pre-RNA. They do not have splicing. Eukaryotes do. At this point, you may want to spend some time going through the objectives from the start of the presentation and just working your way through making sure you can do all of these different processes. This is only the start of protein synthesis. So we've gone from a DNA strand and we have produced a messenger RNA strand from it. And there's another presentation coming which will look at the next stage in protein synthesis, in which the messenger RNA strand is used to produce a sequence of amino acids known as a polypeptide.